Everyone, my name is Wei Liu. I work in the division of Lifecycle API. Today, I'm going to talk about major deficiencies and facility issues in Type 2 DMF. In 2018, FDA finalized a guidance for industry, ANDA submissions, amendments to ANDA under GDUFA. This guidance defines major or minor category for ANDA complete response letter. It also lists examples of deficiencies that FDA may consider major. For DMFs, there are 7 categories in that list. The Division of Lifecycle API established a database to track the major deficiencies in DMFs starting from October 2017. Until now, over 200 first-cycle DMFs have major deficiencies. Within these DMFs, 83% of major deficiencies are due to the qualification of an impurity. Most of them are due to the qualification of a genotoxic impurity. Here are some guidance for qualification of an impurity. The first one is ICH-Q3A. I believe everyone is familiar with this. It defines identification and qualification threshold for organic impurities which are not genotoxic. For genotoxic impurities, we need to follow ICH-M7. When you discuss the control strategies for genotoxic impurities, you are expected to provide a complete list for all genotoxic impurities in your manufacture process. That might include the starting materials, reagents, solvents, intermediates, and any potential byproducts in the process. We have sent a lot of deficiencies to DMF coders because they only addressed some genotoxic impurities, but not all of them. The third one is guidance for industry. It explains how to qualify an impurity in a generic application. If your proposed limit for impurity is higher than the qualification threshold in Q3A, this guidance provides several options to qualify the limit. ICH-Q3C covers the controls for residue solvents. This table summarizes the impurity qualification method and their respective evaluation timelines in DMF assessment. For regular impurities, if you choose USP limit, ICH limit, or set-by-set comparable analysis against the RLD, the DMF assessor can make immediate evaluation. However, if you choose scientific literature or toxic study as the justification, we will send a Pharm Toxic Council to clinical divisions. The timeline is 3 months. In this case, the DMF will be considered major. For genotoxic impurities, if you choose general control options in Section 8 of ICH-M7, the DMF assessor can make immediate evaluation. For any QSA report in the submission, we send this as QSA consult, and it spans 2 weeks. If you choose ICH-M7 Note 4, Note 5, AM's report, or scientific literature to justify, it will be sent to a clinical division as a consult, and the amendment will be major. Similar principles apply to qualification of residue solvents. So, based on the impurity levels in your product, you may keep this in consideration when you establish an impurity limit and its qualification method. Impurity qualification is the #1 reason for major deficiencies. Selection of regulatory starting material is the #2. It accounts for 12% of major deficiencies. ICH-Q11 and its Q&A represent the agency's current thinking on selection of regulatory starting material. When you choose the starting material, all general principles in Section 5 of that guidance should be considered. The presence of a significant structure fragment should not be the sole basis for justification. The concept of starting material in Q11 is same as the API starting material mentioned in ICH-Q7. This explains why the selection of starting material is important, because it represents the starting point of GMP. If you have chosen an inappropriate study material, you may receive our deficiency to ask you to push back. As a response, your initial proposed study material will be considered as an intermediate, which is subject to the guidance on GMPs for API. In addition to categories of major deficiencies we just discussed, there is another situation in DMF, which may make the ANDA application as a major amendment. That is facility. If a required DMF facility is not listed in the drug product application, and that facility requires comprehensive evaluation, the application amendment will be classified as major. In DMFs, there are several scenarios for this. It could be improper selection of regulatory study material. It also could be a reference to a secondary DMF, and the product in that secondary DMF is a critical intermediate. Or you have an outsourced drug substance testing site, outsourced intermediate site. All of these might be the reason you missed a required facility in DMF. Last year, FDA published a Q&A guidance for application 356H4. According to this guidance, the following facilities in DMFs need to be included in 356H4. Final drug substance manufacturer, mechanization, and testing site. It also includes site for intermediate addressed by ICHQ7. If a facility in DMF is required in the application but doesn't appear there, it is called a hidden facility. Let's talk about GMP requirement for API. According to Federal Act Section 501A, all drugs should be manufactured under cGMP. This includes the APIs. ICHQ7 represents the agency's current thinking on GMP for APIs. So API and related manufacture and testing facilities that follow this guidance generally will be considered to comply with GMP requirement. This table is taken from ICHQ7. It gives guidance on the point at which the study material is normally introduced into the process for different types of drug substance. Generally, the steps shaded in gray need to comply with GMP requirement. The requirement of GMP in the manufacture process increases as the process proceeds from early stage to final stage. When we talk about the API study material is the starting point of GMP, we mean the accepted regulatory study material. So if your proposed study material is not accepted by the agency, the GMP starting point in your process will be an early point after you push back the study material. Also from this table, we can see that all intermediate in the manufacture process are subject to GMP. Critical intermediate. Critical intermediate is an intermediate whose manufacture process is considered so important to the quality of drug substance. So its manufacture site needs to be part of facility evaluation for the drug product application. Determination on critical intermediate is made on a case-by-case basis using principles in ICHQ11, as well as the supporting data in submissions. During the DMF assessment, the DMF assessor makes a recommendation based on the manufacture process of the API. The Office of Pharmaceutical Manufacturer Assessment makes the determination if an intermediate facility needs a comprehensive evaluation. This slide shows how to evaluate whether an intermediate is critical or not. Assume we have a manufacture process like this. It starts from the study material A and B. After several steps, it produces the final drug substance G. Here, A and B means the acceptable regulatory study material which complies with ICHQ11. The CT here means chemical transformation. Typically, it involves CX or CC bond formation or breaking. There is a clear relationship between risk and the number of steps from the intermediate to the final drug substance. As the process moves forward from A to G, the criticality of intermediate increases. Specifically, we will consider the following factors when we determine whether it is a critical intermediate. First, overall synthetic scheme and the complexity of chemistry and final drug substance. We will consider how many steps away from this intermediate to the final drug substance. Whether the drug substance quality can be mitigated through the intermediate specification. If there are no adequate isolation steps from the intermediate to the final drug substance, the intermediate will be considered critical. We will also look at whether the process is using common chemistry and reaction conditions or using complex sensitive chemistry. If the submission includes supporting data to demonstrate the impurity carryover has been adequately controlled, the risk will be lower. Otherwise, we consider it higher. Additional consideration is in-process controls and the intermediate specification. For example, an intermediate with purity of 85% and an intermediate with purity of 95% will bring different level of risk to drug substance quality. In most cases, in-situ intermediate doesn't have opportunity to remove impurities because it doesn't have the isolation steps. So we don't consider it as a separate chemical transformation step. However, we also understand that in some real cases, an intermediate is unstable and very active. The chemical reaction is very straightforward. In this case, it's okay not to isolate the intermediate, but please include your justification in the submission. The determination of critical intermediate is based on complete manufacture process from regulatory starting material to final drug substance. When the whole process is covered in one DMF, the regulatory starting material is the material listed in section 2.3 in DMF. When a DMF refers to a secondary DMF, the acceptable regulatory starting material is not the material listed in 2.3 in the primary DMF. Let's continue to use previous case as an example. We assume the secondary DMF covers the manufacture process from A and B to D, while the primary DMF covers the process from D to final drug substance G. Now D is the product in the secondary DMF. It is also listed as starting material in section 2.3 in the primary DMF, but it is not the regulatory starting material in this process when we consider the complete manufacture process from A to G. Actually, it is an intermediate. Whether this intermediate is critical or not depends on the risk assessment from the complete manufacture process from A to G. As we mentioned before, we will consider the complexity of the drug substance, the number of steps from intermediate D to final drug substance G, in-process controls, and the intermediate specifications. Here I want to clarify that whether intermediate D is covered in the primary DMF or in the secondary DMF would not affect the decision. Another point I want to mention is the same intermediate which is non-critical in one process may be considered critical for different manufacture process because we have more than one factors need to be considered. For example, erythromycin A is derived from fermentation process. It is used to manufacture drug substance clarithromycin and azithromycin. Whether this fermentation product is critical or not would be determined by its own specification and its subsequent process controls till final drug substance. It could be considered non-critical for the drug substance azithromycin, but critical for the drug substance clarithromycin. In DMF assessment, hidden facility will be identified in 2 stages. The first one is TCIR process. Here, TCIR stands for Timely Consults and Information Request. This applies for original ANDAs. It happens at very early stage in original ANDA 10-month assessment clock. In TCIR, we look for facility discrepancies between what the applicant lists on their 356H form and what the DMF lists in the DMF submissions. If an issue is identified, the comment will be sent to ANDA applicant in early information request letter. The second one is DMF scientific assessment. This applies for all drug product applications, including the supplements. In the DMF assessment, the DMF assessor identifies required DMF facilities and checks if all the required facilities are included in the application 356H form. If not, we issue the comments to applicant. We have already mentioned TCIR in last slide. This process represents one of the agency's efforts to improve the opportunities for first cycle approvals. It applies for all original ANDA applications and resubmissions under GDUFA 2. TCIR covers 2 areas for DMF. Hidden facility identification is one of them, because new facilities discovered late in ANDA assessment cycle do not allow sufficient time for evaluation and inspection, and may prevent approval. Starting from GDUFA 2, we have already completed TCIR for over 2,400 ANDAs. 15% of ANDAs have a hidden facility issue. This indicates communication needs to be improved on DMF-related facilities between DMF holder and ANDA applicant. How can industry improve? Firstly, please list all required facilities in Section 2.1 in DMF. That includes the final drug substance manufacturer and testing site. It also includes the intermediate site. Then, clearly indicate the responsibilities of each facility in DMF. Please also communicate all required facilities and their responsibilities to drug product applicants. If only certain facilities in DMF are used to support the drug product application, please indicate this in the DMF LOA. Otherwise, the agency considers all facilities listed in DMF applicable to the reference applications. This is a continuous slide of previous one. It lists all required facilities related to final drug substance in DMF. That is, drug substance manufacturers, routine release and stability testing site, mechanization site, sterilization site, storing or warehousing site before the drug substance release. For animal or plant-derived product, it also includes crude extraction site. Also, please be reminded that an alternate site for any of these responsibilities is a required facility. Summary. Qualification of an impurity is the main reason for major deficiencies in DMFs. Following the relevant guidance and choosing appropriate qualification method may help to avoid the major deficiencies. DMF hidden facility identification is another potential barrier for application approval. List all required facilities and their responsibilities in Section 2.1 of the DMF. Communicate early with drug product applicant to facilitate the application assessment and application approval. Determination on critical intermediate is made on case-by-case basis with risk assessment. Appropriate control strategy with supporting data may help to lower the risk. This concludes my presentation. Thank you for your attention. Next, Ashley Boom is going to talk about pharmaceutical quality policies.